Hello everyone, welcome to Get My Class. Let's solve this question here. It says match the following and they've given the tabular column. On left hand side they have given scientist and on the right hand side they have mentioned about the discoveries. And A says Frederick Griffith, B says Michelson and Franklin Stahl, C says Hershey and Chase, D says Jeffries. And under discoveries they have mentioned DNA is the genetic material, then DNA semi-conservative replication, DNA fingerprinting, transforming principle. So they have given different components and we have to match these up, and below they have mentioned A, B, C, D, that is different matches they have mentioned. We have to look for the correct match and finally we have to select the correct option. So let us begin. The first one says Frederick Griffith, and we all know him by his experiment, that is Griffith's experiment. Okay, what experiment he did? He conducted an experiment where he used two strains of pneumococcal bacteria, or you can say streptococcal bacteria, that is R strain, okay, R strain, which was A virulent. A virulent means it does not cause pneumonia, whereas S strain, which causes pneumonia, that is this is virulent. Okay, and then what he did, he conducted various experiments where, like various procedures where S strain actually killed the mouse, whereas R strain does not kill the mouse. The mouse survives. Why? Because it is avirulent, it does not cause disease, whereas S causes disease. Then what he did, at the final stage, he heat killed this particular strain, and when he injected into the mouse survived because it got heat killed, right? So the mouse survived, okay, and there was no disease. But then when he took R strain plus heat killed, okay, this is heat killed S strain, he saw that the R strain after incubation with this heat killed, this is heat killed, okay, heat killed S strain, he saw that this R strain got transformed into S strain and that actually killed the mouse. Okay, so this he called it as the transforming principle, and later he said this is nothing but the genetic material. So the genetic, because of the genetic material, because of the genetic material of the heat killed bacteria, it has been taken up by R, that is R strain, that is from heat killed S strain, it has been taken up by R strain, and that R strain got converted or transformed into S strain. So he said that because it has taken up the genetic material of the heat killed S strain, that's how R strain, that is A virulent strain, got converted to virulent strain, and that is how it killed the mouse. Okay, but he did not know what was the nature of this genetic material. Okay, so he said there is transforming principle and genetic material is the transforming principle. So A will match up with the fourth one. Okay, this is transforming principle. Then the B1 says Michelson and Stahl. So we all know Michelson and Stahl. These people were responsible for proving the semi-conservative mode of replication. Semi-conservative mode of replication. Okay, so this we know very well. So hence option B should be matching up with the second one. The third one, or the C1, says Hershey and Chase. So Hershey and Chase, they did an experiment where they said that actually they continued Griffith's experiment where he said that genetic material was responsible for the transforming principle, but then he did not know the nature of genetic material. So Hershey and Chase, they proved the genetic material is nothing but genetic material here is nothing but the DNA. Okay, so they proved that the DNA was the transforming principle, which was capable of transforming R strain into S strain, and this is the nature of genetic material is DNA. Okay, so this should be matching up with the first one. The D1, Jeffries, he proposed, or you can say he invented a method which is called as DNA fingerprinting. DNA fingerprinting. So we know that DNA fingerprinting is based on the specific sequence what a particular person has in its DNA, because we know that 99.9% of all the human being sequence is the same. Okay, that means the genetic material is the same, but that 0.1%, there is a lot of difference. Okay, so those differences are actually considered for DNA fingerprinting, and there are specific sequences which we call it as satellites, or you can say VNTRs, that is variable tandem repeats, which vary in different individuals, and this is specific for a particular person. Okay, and based on that, DNA fingerprinting was, you can say invented. That particular method was proposed by Jeffrey. So this is the third one. So you can say A is 4, B is 2, C is 1, and D is 3. So option is A is 2, which is totally incorrect. So here itself we can rule it out. Option B says A is 4, that is correct. B is 2, C is 1, D is 3. So this should be the correct answer. Option C says A is 3, which is incorrect. Option D says A is 2, which is incorrect. What is the correct answer? The correct answer is option B. I hope you understood the explanation. Thank you, everyone.